Hello, this is Luke Maffei. I'm currently a research intern at Geisinger. I'm working in the WISE Center for Health and Research. And I came across this article in JAMA that I really took interest in, and I wanted to provide a brief summary for those that may not be as well versed in this health science lingo, and just kind of make a presentation that's a bit more digestible, just to get the general point across and perhaps provide some interesting follow-up information that you may not have known, even if you had read the article. So let's get started. All right, I'm going to go over a lot of terminology these first few slides, just because I want to make sure everyone can comprehend what I'm saying. And again, I want to make sure that I can comprehend what I'm saying. I'm still relatively new to this, so if throughout this presentation you notice any sort of discrepancy, or perhaps I didn't make myself clear enough, sound off in the comments, or shoot me an email. I'll put up an annotation of some sort to correct my mistakes. Anyway, first thing I want to cover is a GWAS. A GWAS is a study that involves examining the different genetic information from different patients. The goal of these GWAS studies are usually to compare individuals with varying alleles, but they have a similar condition, yet they express different phenotypes. So an example of this could be a study I read recently. It was relating those who had skin cancer with pigmentation, and they ended up finding, if you carry genes related to albinism, then you're more likely to be at risk for getting skin cancer. But that's just one example. It's basically taking a group of subjects that have some sort of trait in common, but there could be a different phenotype. It could be a different reaction to a drug, like we're seeing in this case. It could be a prolonged period of recovery. ALL, aka acute lymphoblastic leukemia. It's also called acute lymphocytic leukemia. It is the most common childhood cancer. It's brought about by rapid, unwarranted division of white blood cells or lymphocytes. St. Jude recently reported that approximately 90% of children diagnosed with ALL can be cured with proper treatment. A decade and a half ago, this number was only about 80%. So methods, I'd assume, have gotten a lot better as far as treating ALL. A much higher success rate, a notably higher success rate. So that's some good news for families that may have a child with ALL. The treatment's there and it's getting better. Vincristine is one of the drugs used during chemotherapy. It's a Vega alkaloid. There are some others. Vinblastine, that one's used for a variety of different cancers, such as brain cancer is one. Vinorelbine, that one's more associated with treating lung cancer, but the one we're concerned with right now is Vincristine. Just throwing out that Vega alkaloids, they're a very common agent used in chemotherapy. Basically, these Vega alkaloids interfere with microtubule formation, meaning that the meiotic spindles that kind of help the cell divide cannot properly form, and that leads to apoptosis. Side effects of Vincristine, however, include constipation is probably one of the most common, motor weakness, and what's of interest in this study was peripheral neuropathy. There are a bunch of others, like some experience problems in their digestive tract, or in addition to constipation, they have problems passing urine. So Vincristine, though it's an effective treatment most of the time, it can also have some unwanted side effects. Here we just have a brief overview of the phases of mitosis. What I really like about this image is when we look at metaphase and anaphase in particular, you can see those meiotic spindles attaching to the chromosomes. When Vincristine is used as an agent, however, they can still form, but not to the degree that they'll be effective. I'll go into more detail in the next slide. So you're basically disrupting the cell's ability to divide, and if the cell cannot divide, that cell will die. Here's a brief overview on how Vinca alkaloids work. Just so you know, most of the graphics in this presentation are ones that I borrowed from different sources, but there are a few that are mine. If there's no credit underneath, it's probably mine, unless it's the title of an article. What we have here, the vinca alkaloid acts by capturing free alpha-beta tubulin dimers and forming aggregates. I'll be honest, I'm not quite sure what it does as far as interacting with the gamma portions, but vinca alkaloids, essentially, they're going to kind of isolate and aggregate these dimers and prevent them from forming the microtubule and rather form an alternative structure. Again, microtubules can still form, but not to the degree that they'll be effective. So this vinca alkaloid, it will basically cause the cell to kill itself. The thing about peripheral neuropathy, there's such a huge scope as to what this means. There are many different severities of peripheral neuropathy. It could be numbness in your fingers and toes. It could be your ability to perform certain motor functions may decrease, like you may not be able to button your shirt or eat with a spoon or tie your shoes. Or in some severe cases, it can cause paralysis and possibly death. So when I say peripheral neuropathy, just know that I'm referring to an entire scope of this sort of disorder, not just one scope in specifics, unless I mention it. But symptoms range from pins and needles sensations to sharp pain and weakness. And it's weird because as far as pain goes, you may have decreased pain, you may have increased pain. So it's very dangerous in that, you could be overreacting to a certain stimulus, or you may not be reacting at all. I'm just using the term SNP because it's what was used in the article that I'm addressing. SNPs can occur anywhere within a gene, a gene's promoter region, an intronic region, etc. Essentially, for each SNP, there are different alleles that aren't always, well, more often than not, aren't common from person to person, because these alleles vary with their base pairings. So I could have a T where someone else would have a C. That's basically what a SNP is. One of the SNPs in particular that was of high interest in this experiment was found on chromosome 5, near the very end of the P region. It's RS924607. The alleles for the SNP are C and T, and it falls in the promoter region of CEP72, that's centrosomal protein 72. Also of interest, if you notice inside the little graphic I have on the right-hand side of the screen, you'll also notice to the right of CEP72, you'll see TPPP. That's tubulin polymerization promoting protein. Again, doesn't have much to do with RS924607, but the funny thing is I actually did a little bit of research on that gene in the past. So it was just very, I don't know if you want to call it by luck or some sort of weird coincidence, but there it is. All right, this what I have right here is a Manhattan plot. In layman's terms, because I got to be honest, as far as my understanding of this plot goes, it's way beyond me. There is a certain threshold that a SNP needs to reach to be of statistical significance in a GWAS. And as far as RS924607 goes, it was the most significant. The second closest you can see is on chromosome 2. That one, I believe, oh my gosh, I hope I'm not messing this up. It's RS17032980. I'm pretty sure that's what it is. That one's more associated with, came up in a study on schizophrenia. So again, when we look at the ones that are more relevant, it's the most significant SNP is, by far, RS924607. Okay, CEP72. I probably mentioned it before. The CEP72 gene codes for the CEP72 protein. And the CEP72 protein plays a critical role in redirecting other proteins to the centromere. And without that, the meiotic spindles probably couldn't form as efficiently. That was an email. That wasn't part of the presentation. Just know that CEP72, the gene codes for the CEP72 protein. Without that protein, cell division would not be able to occur as efficiently. Oh, also CEP72, it's part of the leucine-rich repeat superfamily of proteins. Leucine, amino acid, it's in there a bunch. And These proteins tend to be more horseshoe shaped. All right, so the main issue. Finally, we're to the meat of the article. Though it's effective, vincristine can have negative side effects, such as peripheral neuropathy, which is kind of a big deal. Though these cases are usually mild, there are times that they can be fatal or life-threatening. Like I said earlier, if the subject gets paralysis, that could be a major issue. And prior to this study, there was no substantial method of predicting whether or not a child being treated with vincristine would have a more severe reaction when being treated with the zinc alkaloid. All right, so the St. Jude study, first of two studies I want to get into. This is the main one, as I was mentioning before. The second one, I'll get into that a little later. It was a prospective study, 321 subjects from two separate cohorts. As you'll see later, sample size kind of decreased as the study went on. I'll explain that in detail in the coming slides. The objective was to identify genetic germline variants associated with the occurrence and severity of vincristine-induced peripheral neuropathy in children with ALL. And they had to run the GWAS to see if there was any genetic variant that had a higher frequency of peripheral neuropathy prompted by the vincristine treatment. So that Manhattan plot I showed earlier, they didn't have that prior to this study. That was inclusive in this study. I believe I found that in the supplement. All right, subjects. Now we're getting into more demographical information. There were two cohorts, each done at different times, approximately 10 years apart. The first one was the St. Jude Total 8B, I believe that's what it's supposed to mean. Yeah, the 8B protocol, enrolled in the 90s. Now here's something significant. The median age was 6 years, and they had a substantially larger patient size. When we look at the Children's Oncology Group, I'm just going to call it COG, it takes place approximately a decade later, and the median age, this is something of interest, it's much higher, like significantly higher. And something you have to remember, commonly in these childhood cancers, the younger the child is, there tends to be a higher success rate in treating that cancer because, a younger child will have a higher metabolism, and their response to chemotherapy is usually a bit nicer. And this cohort, the COG cohort, had less than half the amount of patients. And over here, as far as the characteristics go, majority of the patients were male in each study, and the majority of patients were also European. But there were a significant amount of patients of African and Hispanic lineage. Now, as far as each of these protocols had 3 phases, but they were significantly different. Inside the St. Jude protocol, we had 8 weeks of induction, 2 weeks of consolidation, and 120 weeks of continuation. Compared to the other study, that's a lot longer for the continuation phase. Each patient was scheduled to receive 36 vincristine doses, but the concentration of those dosages varied. They were between 1.5 and 2.0 milligrams per meter squared. Over here, I just have a quick flow chart, just kind of displaying why we lost some of the participants. It's not necessarily implied that people died. It's just essentially demonstrating that we couldn't get the DNA from everyone, and then the genotyping failed for 20 people. But you got to take into account this was a study done, I'd say approximately 2 decades ago now. So, for all we know, maybe perhaps their genotyping technique wasn't as sophisticated as what we have today. I couldn't even say the word sophisticated. What kind of researcher am I? Looking at the Children's Oncology Group, we have 3 phases again. What I really want to point out, the 50 weeks of maintenance therapy, that's essentially kind of like another term for continuation, that is less than half the amount as the other one. But regardless, they still received a comparable amount of vincristine doses, 39, just 3 more. And this group was divided into 2 separate groups, which, as you'll see, was somewhat unfortunate. Group A received approximately the same dosage as the other protocol mentioned before, but Group B received significantly more. Now remember this, significantly more of a dosage of vincristine apparently as displayed by this, will most likely lead to a higher rate of Vincristine-induced peripheral neuropathy. And Group B was eliminated from the study. They had to treat these patients for neurotoxic effects later on. So unfortunately, as we can see in the next slide, Group B, they're out of there. They're not part of this finalized study. And if we look over here, genotyping failed for only one in Group A, and this was done a bit more recently. So hey, who knows? Maybe like I said before, our genotyping techniques, maybe those improved. I hope they would in 10 years. Okay, evaluation. This is just something that I thought I'd point out because we're going to be looking at graphs later. It might explain why some don't look as pretty as other ones. Throughout each study, participants were evaluated by oncologists for presence of peripheral neuropathy via physical examination. In the St. Jude cohort, they were assessed weekly throughout all phases of the study, but in the COG cohort, they were assessed weekly for the first year, but then after that, they're only assessed monthly. And we'll see that later on. You'll notice that the graph for St. Jude, it's a bit more smooth, I want to say? I don't know, you'll notice later on. Common terminology criteria for adverse events. This was used to, when the oncologists were evaluating the patients, this was to kind of place a grade on their condition. Because as I mentioned before, peripheral neuropathy, there's a huge scope. It could be anything from I can't feel my fingers to I can't move my arms. The grades I have in greater detail over here. Again, this one was taken from the supplement. The more severe grades, it'd be 3 and above. Luckily, there are no grade 5s reported. No one died. And there was actually only one occurrence of grade 4, paralysis. But you'll notice a lot of the analyses to follow, grade 1s tended to be omitted because like they still experienced some sort of effect, but it was nothing crippling. Because inside grade 2, you couldn't put on your shirt, or you couldn't write, or some people had trouble speaking. Grade 2 is when your quality of life is actually being significantly affected. I'm not saying that those with grade 1, weren't in pain of any sort, but it wasn't quite as significant. All right, genotyping. 500 nanograms of germline DNA was isolated from normal peripheral white blood cells before they were digested with a restriction enzyme, and the DNA samples were amplified, labeled, and hybridized through various techniques. SNP imputation was carried out. There was a total number of 21 million SNPs, and the race was determined genetically. What I assume from this is if I am 1/16 Hispanic, but I am 15/16 Caucasian, in this study I'd most likely be labeled as Caucasian. Cellular analyses. Okay, primary ALL cells were isolated by applying Ficul hypoQ, I probably said that wrong, gradient. These primary ALL cells were tested for vincristine sensibility in vitro using an MTT assay. In vitro just means outside the body. If it was in vivo, it'd be inside the body. Sensitivity to vincristine was measured by using a concentration lethal to half of these leukemia cells. So that's kind of how they determined what the dosage would be. Ficul hypoQ gradient. Gosh, it's like I'm speaking another language. The one thing I want to point out about this graphic, when I say blood on the before, sometimes, well, from what I've read, the blood's actually diluted. Don't take my word on that. I've never actually ran one of these. But looking at the after, our main concern, what we want to isolate are those white blood cells. Red blood cells don't even have a nucleus. Who cares? Plasma, that's just superfluous liquid. At least in this experiment it is. So we wanted to get those white blood cells isolated. I'd assume that this was done like through some sort of a centrifuging technique. Don't quote me on that. Don't quote me on any of this. I'm very new to the field. I'm just sharing what I know. Statistical analysis. Associations between SNP genotypes and phenotype of peripheral neuropathy were made by establishing a weighted logistics regression model fitting for recurring events. Something else I mentioned down here, specifically, as I mentioned before, the researchers were concerned with the number of occurrences that were classified as grade 2 or higher. So the ones that were more significant on affecting the patient's quality of life, or just, their ability to perform daily tasks without assistance. The relationship between the cumulative vincristine dose at the first toxic episode and the genotype was analyzed, as well as the relationship between severity of toxicity at the first episode and genotype. A linear regression model was used to test the association between vincristine sensitivity and the SNP genotype in primary ALL cells. If you're wondering why I'm just reading straight off the slide, honestly, as far as statistical analysis goes, statistics isn't exactly my forte. As far as interpreting this and conveying it to you in a manner that both you and I can understand, I'd recommend just reading the article if this is what concerns you. Honestly, I'm no expert on this sort of thing. All right, results. In the St. Jude cohort, grade 2 to 4 vincristine-induced peripheral neuropathy during continuation therapy occurred in 64 of the 222 patients. And in the COG cohort, as you see, there was slightly less as far as grades 2 to 4 went. Only 22 of the 99%, 22.2% approximately. There's a significant difference, but the sample sizes were relatively smaller. If we had like 1,000 kids in each study, these numbers would probably be a lot closer. All right, comparing genotypes. 20 of the 32 patients with the TT genotype in the St. Jude cohort developed at least one episode of the vincristine-induced peripheral neuropathy. Later on, I'll explain a little more what this TT genotype means. And then there was 44% in the COG cohort. In total, over 50%, 56% with the TT genotype developed at least one episode, grade 2 to 4. But when we compare those to those that have the non-risk genotype, aka CC or CT, it's less than half. It's only 21.4%. All right, moving on. This is just a table. What I really just want to point out is when we look at grades 1 and 4, there's significantly less. Many times, if a patient did suffer peripheral neuropathy, it was somewhat crippling, but it wasn't necessarily life-threatening, because we only have one instance of a subject being paralyzed for grade 4, and that one wasn't even with the risk genotype, might I add. When we're kind of observing this, I guess we can just kind of make the assumption that the dosages that were given, they weren't, aside from Group B in that last study, I don't have any outside information on them. As far as the dosage that was given to these patients, it seems like the protocol they were following, they knew what the maximum dosage should be in order to prevent a life-threatening condition such as paralysis. Something else just to point out, in both studies, the risk genotype, the TT, was the patients with that risk genotype, they were the ones that had a higher occurrence of this vincristine-induced peripheral neuropathy. And as far as CC and CT go, there is a slight difference in both cohorts, but that's not really as concerning. Our main focus here is the comparing the TT genotype to the other two, because TT, as we can see in both, having that genotype increases your risk for this vincristine-induced peripheral neuropathy by 100%, twice as much. As I mentioned before, the reason why the graph on the right may not look as nice is because they were only evaluated monthly as opposed to in the St. Jude cohort when they were evaluated weekly. So we have 4 times as many measurements in the graph on the left rather than the one on the right. So that's why it looks a bit more rounded. You can kind of see the trend. It doesn't necessarily plateau, but the highest rate of this increasing number of children that suffer from peripheral neuropathy seems to have occurred around the first half year. Moving on, the one on the left is simply a combined graph of the last two I just showed. The one on the right, that's the cases classified as severe. That'd be grades 3 to 4. Well, as far as them throwing 4 in there, I guess they just did it to be more precise as far as what they classify as severe. But honestly, if they put just grade 3 on there, it probably would have been sufficient, because there was only one case of grade 4, and that wasn't even inside the group that had the risk genotype. All right, you've heard me say it 1,000 times. Now's when I'm actually going to elaborate on it a bit more. The CEP72 risk allele is the T at the SNP discussed earlier, RS924607. when you have this risk allele, there is reduced CEP72 expression in the ALL cells. These include ALL cells of both T and B cell lineage. When you compare it, and this is a fact I got off Snippedia, brilliant source by the way, check them out. When compared with those with CT or CC genotypes, those with a TT genotype have twice the risk, approximately, of developing this peripheral neuropathy when treated with the vincristine. All right, this is a visualization of what I mean when I just want to get across what I mean when I say allele, when I say like a T or a C allele as opposed to a genotype. There are two locations on each chromosome that you'll find this SNP at, one on each chromatid. At this location, if your base is, the major is a C on both, you're fine, you're common. If you have only one risk allele, you're still considered common as long as you have one of those Cs. However, if you have that risk allele on both of the chromatids, then you're at risk. If you have a TT genotype or two T alleles, then you are at risk for, you are more at risk, I should say, at acquiring peripheral neuropathy or even perhaps a more severe type of peripheral neuropathy after being treated. And again, this was in subjects that had ALL. As far as other ones, I'm not sure if I can quite speak on their behalf. As I mentioned earlier, I mentioned that there was a variety of ethnic groups involved in this study. And something really interesting about this study is it actually kind of explained a phenomenon that was being observed. They noticed that those of African descent did not have as many episodes of peripheral neuropathy, or the severity was not as high. Well, when we look at the HapMap populations, especially the Kenyan populations, the T allele frequency is much, much lower. Like if we look at the two Kenyan sections, for each it's like less than 10%. So a significant difference, and that explains why. That explains why those of African ancestry often do not have as severe a reaction to vincristine as far as peripheral neuropathy goes, because the T allele is less common. All right, this was found in the supplement, but I still feel it's worth noting because it kind of goes a bit more in depth on why this TT allele can be dangerous. The presence of this risk allele causes a stronger binding affinity to NKX 6.3 homeodomain, and increases the binding affinity 400,000-fold, which is, crazy. And this was confirmed through electrophoretic mobility shift assay. So the greater binding affinity results from this risk allele in the promoter region for CEP72. And when this occurs, CEP72 expression is significantly lower. All right, the big picture, if you're going to take home anything from watching this video, at least take home this. From the study, we can assume that those that have this risk allele in their CEP72 promoter region have a significantly higher correlation with increased vincristine sensitivity and induced peripheral neuropathy. I have a nice little graph over there. In both cohorts, the TT genotype, those that displayed it, had a higher amount of occurrences of this peripheral neuropathy. All right, even though this study's brilliant, it was published in JAMA, and JAMA I know has very high standards. There are a few limitations. There was a small sample size, and out of that sample size, less than a sixth of the participants had this TT genotype. And then the other shortcoming I made note of was COG Group B, because they gave the kids too much vincristine, or the dosage was too high, and these children needed to be treated. Again, it was nothing purposeful, but it was still a negative outcome for this group. But despite the shortcomings, I found the study to be conclusive. I really thought the main article is brilliant, and if you want to do further reading, the supplement is more than enough. Just really goes into detail on a couple of topics that they just kind of glanced over in the article. But I understand, you can't publish an article that's 50 pages long. I understand why it needs to be shortened. this is the other study I mentioned earlier. This one's more recent. And the interesting thing is it makes constant reference to the last study I mentioned. And to my knowledge, the full study is not published in English, but it's very easy to translate just with I just use Google Translate. So maybe that's why I haven't heard as much about this. Granted, it was published in Pharmacogenetics and Genomics. Well, not the entirety of the article, but kind of a synopsis. And also, it was published in Pediatrics Week. I didn't mention that on here because it's a smaller publication. This one in particular was in Spain, and I'll just switch over to the major differences. It has smaller sample size, only Spanish Caucasians. Dosage was less, like just about half as much. And rather than studying later phases after induction, such as continuation, they were mainly only interested in the induction phase and the occurrences of peripheral neuropathy. And as we see with the results, I'm just kind of glancing over this, they didn't really find the same correlation or the same association as the last study with that risk allele and the TT genotype. In fact, if anything, they found a lower occurrence, which is somewhat odd. And again, there are multiple reasons to explain this. I'll go on to that on the next slide here. Both studies had small sample sizes. There was a difference in the gene pool because, one was done, the St. Jude study was done more domestically. This one was done in Spain. There were differences in the Vincristine doses. Perhaps if the study in Spain used higher dosage, maybe they would have most likely they would have more cases of this Vincristine-induced peripheral neuropathy, and perhaps that could, slant the results a little bit more towards what the St. Jude people found. And this is the most significant. They were studying a whole nother phase. They were studying the beginning of the phase as opposed to towards the end, or the after the beginning. Why did I include the Spanish study on here? First off, I'm not trying to discredit JAMA or the St. Jude study at all. I think that's a fantastic study. I think it's worth reading. It's that I think this study serves more as a supplement than an argument against the previous article I mentioned. The Spanish study shows how the protocol differs for treating a child with ALL. With Vincristine, the dosage in Spain is much lower, significantly lower, at least in this study. I can't speak for the grand scheme of things. And then there were a bunch of parallels with the St. Jude study, which makes sense because they even mention the St. Jude study by name. They both use the same criteria for grading the patients. They both show that the TT genotype is the least common in both regions. And there are other correlations, such as they had more male patients than females. So we can still learn quite a bit from this Spanish study when we look at it as more of a supplement than a disagreement. Because you got to take into account, they didn't recreate the previous study. They had their differences. Finally, this is something that I feel is really worth mentioning. The fact that this study was done in Spain, it just kind of paints a picture of how significant this issue is. ALL, it's the most common childhood cancer. And the studies, not just nationally, not just here in the United States, but they're being done worldwide. This Spanish study just, serves to credit that statement. There is legitimate interest in, improving techniques used to treat ALL. And it's important that if we are to make any progress, we all need to work together. All right, finally, this is just my works cited. I apologize if this was super lengthy, and I'm sure I screwed up at least a couple things somewhere throughout this presentation, but I do hope you learned something from it. And this is the first time I've ever really done anything like this. I'd love to do more presentations like this. It's just, what bothered me is when I was searching this topic on Google, YouTube, what have you, ProQuest, I wasn't finding that much, so I just kind of wanted to create a digital presentation that people could watch, because I can understand when you're reading through these scholarly articles, they're chock full of information, but the question is how much of that information is relevant to what you're looking at, and how much of it do you actually understand? Because I understand the language and some of the nomenclature can be, a little hard to digest for, a high school student, or hell, even a college student. I honestly had to look up a couple of words. So I hope you learned something, and I look forward to doing more things like this in the future. So yeah, just sound off below in the comments if I've really screwed up anything, and yeah, take care.